Okay, so welcome to the second video in chapter 3, where we are going to continue our discussion on cells. So we've already talked about the general characteristics and organelles found in eukaryotic animal cells specifically. What we're going to look at now is how molecules and other ions like water, salt, those types of ions and molecules move in and across the plasma membrane. There are a couple of different ways that this can happen. One is called passive transport. Passive transport is a type of movement that does not need energy for those molecules to move. So the cell can allow these molecules to move in and out of the cell, and it doesn't use any ATP during this process. So no energy is needed. We're not using resources to move these substances across the membrane. And the typical kind of processes that are governing passive transport are diffusion and osmosis, and we'll define what those two processes are here in a second. The second type of movement is active transport, and this is a type of movement when cells need a product to be either outside the cell or inside the cell, but they have to spend energy. They have to use ATP in order to move that product or to move that molecule. And this is where our cells use ATP in order to make that happen. So this is where they're expending energy in order to move that product. Then we have a third transportation mechanism, which is referred to as bulk transport. This is where you have that vesicle to move larger substances into or out of the cell. I mentioned these terms earlier. So into the cell is endocytosis, and then out of the cell is exocytosis, right? So this would be your secretory vesicles, right, that we talked about earlier. All right, so now we're going to just look at each one of these in kind of more detail. So first, we're going to talk about passive transport. Passive transport, like I mentioned, uses no energy to move a substance, and it does so based off of diffusion and osmosis. So basically, diffusion is when molecules will move from an area of high concentration to low concentration. And one way to think about diffusion is if you have a bowl of water and you drop food coloring or dye in that water. If you just do one drop, you know that that drop will hit the water and it will be concentrated in that one spot where that drop hit. Now, because there are no dye molecules around that drop, it will start to spread out and diffuse into the water. So it goes from the area of high dye molecule concentrations where that drop was to area where there's low dye molecule concentrations, which is everywhere else. And so it spreads thin. And so we say that molecules will then move down the concentration gradient. So they're basically going from high concentration to low concentration. So that's down the concentration gradient. Okay. And so here we have an image, right? It's just kind of showing this diffusion with dye in a gel. And so they have put and concentrated the dye here. And then over a day, you see how those molecules diffuse outward, right, in both directions. And again, this is going from a high to a low concentration gradient. So let's see. Yeah, we'll talk about that in a second. Okay, so that's diffusion. Okay. Next is osmosis. And so osmosis specifically refers to the movement of water, so the diffusion of water. Okay. Now, osmosis can be a little tricky to get down because of the way it's worded and how the process happens. Right? So osmosis is the diffusion of water across a selectively permeable membrane. And what we mean by a selectively permeable membrane is that this membrane will only allow water molecules to move across it. No other molecules can move across this membrane. And so like salt, sugar, right, those molecules are not able to move across the membrane, so it's only water. And so the way the water moves is that it diffuses from a low solute concentration to a high solute concentration, right? So this is the opposite of what we just said. So it goes from low to high. But remember, one thing I want to point out, this is the solute concentration. It's not the water concentration, okay? We're not saying water goes from a low water concentration to a high water concentration. It goes from low solute concentration to high solute concentration. And remember from chemistry, right? Solute is what is dissolved in the water, right? So this could be something like salt or sugar, right? And so what's going to happen is if you have water that has no salt in it, and that comes into contact with water that does have salt in it, and there's a selectively permeable membrane, the water with no salt will move into the area of water with high salt because it has more solute in the high salt, right? So higher solute concentration. We'll show an image of this in the next slide to hopefully clarify any confusion. Now, the result of the pressure that happens to oppose osmosis is the osmotic pressure. Okay, so it's basically the pressure required to oppose that osmotic force. And so if we would show this, right, this is a typical example of the process of osmosis where you have this U-shaped beaker, you have the water level, okay? On the right-hand side, right, there's no solute. So these little green molecules are solute molecules. In this case, it's sugar, it's glucose molecules. So what this means is that there is a low solute over here. There is a high solute over here. And so that means water wants to move across this selectively permeable membrane here in the direction of low to high, okay? So water will move from right to left because it will move from low solute to high solute. And so what actually happens is the water level will decrease, right? So it goes down, right? As the water moves across over here. And the remaining difference, right? So the difference in the water level now would have been the pressure that was needed to oppose the water moving in this direction. So that difference is the osmotic pressure. Okay? So osmosis again is low to high, right? Solute concentrations. Water will move from low to high solute concentrations. Okay, so that's the key thing. And remember, this membrane is only allowing water to move across. Okay. And now, just to answer this question, in case you may have it, you may be thinking about it. Well, what would happen with the sugar molecules if they could move across this membrane? If these sugar molecules could move across this membrane, then the sugar molecules would move and diffuse in this direction because they would go from their high concentration to their low concentration, right? So they would even out if they could move across. Okay, but since they can't move across, water is going to compensate for that, and osmosis is going to happen. Okay. And now remember, these two processes, diffusion and osmosis, are a type of passive transport. And so they're moving with a specific concentration gradient. And so diffusion can allow molecules to move directly through the phospholipid bilayer, the plasma membrane. But only certain molecules can do that. So these are small molecules and they have to be uncharged, so meaning nonpolar. Because remember, those fatty acid tails, they don't like polarity. They don't like any molecules that are polar. So if there's a polar molecule, then it's not going to be able to diffuse. It's going to have to move through using a protein channel. Now, if it's a bigger molecule, so like a molecule of water or ions, which are charged molecules, they still can diffuse through passively, but they have to via a protein channel. Okay. So those proteins that are embedded in that plasma membrane allows them to move across. Then you could also have what's called facilitated transport or facilitated diffusion. This is where you have a protein that changes shape and transports the molecule through a bilayer. And so because it has this shape recognition, these are highly specific molecules that are going to travel through. So if it's just like a diffusion channel, open, say like pipe, which is the protein that moves through the plasma membrane, anything that can fit in that pipe or opening can move through. But if it's a facilitated diffusion, only the molecule with a certain shape can move through that protein channel. So for example, glucose will move through a facilitated protein. It will undergo facilitated transport because that protein will only move glucose because it actually has a shape that only fits glucose. Okay. So here's a figure where we are showing these three type modes of transport, passive transport. So here you have all of the molecules on the outer portion of the cell. So this is the extracellular environment up here. Notice that all of the molecules are in a higher concentration on the outside versus the concentration on the inside. So that means they all want to move outside to inside. And so if the molecule is small and it's nonpolar, then it can just kind of move through the plasma membrane. So these spaces in between the phospholipids. However, if it's a little larger, or let's say these are positive or have charges, they can't move through because these tails don't like those charges. So they have to go through this channel. Right? So it's just the opening that allows those larger charged molecules to go through. And then here on the right, we have that facilitated transport. So you have this glucose molecule, large, has a specific shape. Its shape only fits in this protein. As it goes in, it attaches to this binding site. The outside closes shut. The inside will open, and then that glucose molecule will be pushed out into the interior or intercellular space and go inside the cell or outside the cell. Okay, and so these are the three kind of mechanisms of passive transport. And it's important that you really learn these mechanisms of transport, both passive and active, because we will refer back to passive and active. We will be using these terms all throughout the semester because we'll constantly be talking about something that's, diffusing across cells into the bloodstream or into the liver or into the kidneys or from the lungs into the bloodstream. So lots and lots of active and passive transport talk is going to happen in this semester. Okay, so now after passive transport, we're going to talk about active transport. So passive transport is moving molecules and ions with no energy. Active transport is moving these substances with energy. And active transport needs energy because the cells are moving these substances against their concentration gradient. So for active transport, normally, these molecules are going to move down their concentration gradient, so from high to low. But for when active transport, usually the cell is moving it from a low to high concentration. Um, and because they're being moved against the concentration gradient, they need energy to do that. One of the best examples I can give for active transport is for those of you that have children or have ever babysat. You tell your kids to go to bed. Sometimes they don't want to go to bed, right? So they're not passively going to bed for you. Um, you have to get up, spend energy, pick them up, move them, and put them in the bed, right? And so you're actively putting them to bed. This is basically the same thing. So the molecules can move into or out of the cell as long as they're moving down their concentration gradient, so passively. But whenever you want to move them in a way that they don't want to go, like kids not wanting to go to bed, you have to put energy to move them yourself against the direction that they want to go. And so the ATP provides the energy that pushes those molecules against their concentration gradient. One of the most well-studied active transport mechanisms is the sodium-potassium pump, and we will talk about this pump. We'll go into a lot of detail about this pump, and it's just a very important pump in our body because it helps to regulate cell volume um, and keep a state of homeostasis in terms of cell volume and the amount of water that is required inside of our cells. So here's an image of active transport. So you have these molecules, right? Notice you have high concentration versus low concentration. And so the molecules passively want to move into the cell, but there's no way for them to move into the cell, so they don't move into the cell. However, the cell wants these molecules out. And so what happens is there's a protein channel, right, that is selective for that molecule. That molecule will go into the protein channel. ATP will attach to it. The energy causes the shape change to push that molecule out and move it against its concentration gradient. Okay. Now, another way that molecules can move against their concentration gradient is through carrier proteins. So carrier proteins will use the energy of another molecule moving to move a molecule against its concentration gradient, right? So let's change the color on our pen here. Let's get the same color. So we have low concentration gradient for the squares, high concentration gradient for the squares, and we have a low concentration gradient for the spheres and a high concentration gradient for the spheres. So the direction of movement wants to go this way for the spheres and this way for the squares. The cell wants the spheres on the inside, but it also wants the squares on the inside. The squares are wanting to move out and go with their concentration gradient. So the cell can take advantage of the concentration gradient of the spheres because it's going to passively move in. And that passive movement is a form of movement that's generating energy, right? Because it's moving, right? That's kinetic energy. And it can bring that square molecule into the cell without having to spend the ATP. It's basically piggybacking on the sphere. So the sphere is moving through passively, bringing energy with it, right? Energy of that movement. And the energy of that movement with the sphere is being used to bring the purple molecule, the square molecule, in against its concentration gradient. Right? So in a direction that it doesn't want to go. And again, that's called a carrier protein. And it's transporting that molecule against its concentration gradient. We have a little video here on active and passive transport. Make sure my sound's shared.
Passive transport requires no expenditure of energy. In simple diffusion, water molecules and gases like oxygen and carbon dioxide need only move down their concentration gradient to move into or out of cells. Molecules that are larger than oxygen but that are fat-soluble also move into the cell in this manner. In facilitated diffusion, transport proteins are required for adequate amounts of water and polar molecules larger than water to pass through the plasma membrane. They are a hydrophilic passageway through a hydrophobic environment. Glucose and other hydrophilic molecules, such as amino acids, move through the plasma membrane in this way. This form of transport does not require the expenditure of energy because it also has a concentration gradient working in its favor. Passive transport requires no expenditure of energy, but active transport does. In active transport, molecules again move through a transport protein, but now energy must be expended to move them against their concentration gradient. A cell moves solutes against their concentration gradients by using pumps. One of the most important and thoroughly studied of the active transport mechanisms is known as the sodium-potassium pump. It works in the following way. First, 3 sodium ions located within the cell cytoplasm bind with the transport protein. Then, the energy molecule ATP gives up an energetic phosphate group to the transport protein. This binding causes the protein to open its channel to the extracellular fluid. The protein loses its sodium ion binding sites, thus releasing the ions into the fluid. Binding sites are then created for potassium ions. 2 potassium ions move into the protein's potassium ion binding sites, which brings about the release of the phosphate group. This loss returns the protein to its original shape, releasing the potassium ions into the cytoplasm and readying the protein for binding with another set of sodium ions.
Okay. And so that shows, active and passive transport, energy versus no energy. But it also gives a really good diagram on the sodium potassium pump. Basically explains it for you. But we'll go over it in a little more detail later on, where we focus on how it helps regulate specific processes and also cell volume. The last thing we want to talk about with transport is the bulk transport, so endocytosis and exocytosis. So remember, with endocytosis, that's vesicles bringing substances into the cell. And so as it enters the cell, it's surrounded by a membrane, and that membrane basically it kind of invaginates and pinches off, and then brings that stuff, the molecules, into the interior portion of the cell. Whereas exocytosis, it gets rid of stuff from the cell, so it expels them from the cell. And it's basically where a vesicle that already has material inside of it fuses with the plasma membrane, therefore pushing those substances into the external or extracellular environment. So here we have 2 images that's showing that happen. So the top one is endocytosis, where we have the plasma membrane kind of invaginating. Those molecules are being captured, and it pinches off and buds off of the plasma membrane and creates that vesicle that's going to transport that material, those substances, into the cell. And then the opposite happens with exocytosis. You have a vesicle with substances already inside. It goes to the plasma membrane, it fuses with the plasma membrane and pushes those substances into the extracellular matrix or extracellular fluid. And so again, we have an animation just kind of showing those processes.
The two mechanisms for moving big particles through the membrane are called endocytosis and exocytosis. Exocytosis is the movement of particles out of the cell, while endocytosis is the movement of particles into the cell. During endocytosis, relatively large materials are moved into the cell by the infolding of the plasma membrane. There are a few different forms of endocytosis, such as pinocytosis and phagocytosis. In pinocytosis, literally cell drinking, the plasma membrane forms a kind of harbor that pinches off and moves into the cytoplasm as a vesicle. The vesicle carries primarily water and some solutes. In phagocytosis, literally cell-eating, larger materials such as food particles or bacteria are enveloped by pseudopodia. The pseudopodia surround the particle and their membranes fuse together, forming a vesicle that moves into the cell interior with the captured particle enclosed. During exocytosis, materials are moved out of the cell through a fusion of transport vesicles with the plasma membrane. The transport vesicle moves toward and attaches to the membrane, releasing the vesicle's contents into the extracellular fluid.
Okay. So not only can substances and materials pass through the plasma membrane, um, the plasma membrane can also serve as a mechanism for information and communication to happen between cells. And so there are not only transport proteins in the plasma membrane, but there's also receptor proteins. And these receptor proteins are required for the transmission of information, and that's either to or from the cell. And on these proteins, there are specific sites that only can fit certain receptors and certain shaped molecules. And those signal molecules will come in and they'll attach to those receptor sites. And when they attach to that receptor site, um, there's a change that happens, and there is some sort of um, cellular process or reaction that happens at a response or as a response to that signaling molecule binding to that receptor site. So, for example, you have your receptor protein here, the signal molecule coming in and binding. You have a specific substrate, and that substrate is turned into another molecule, so a different product, due to the chemical reaction that happens as a result of that signaling molecule binding to that receptor site. And then that signaling molecule will kick out and go on, and then more signaling molecules can come in and trigger that chemical reaction again. And so the body can release certain signaling molecules, say when it needs more of a particular product made, or it can not release those molecules, those signaling molecules, if it already has enough of that product and doesn't need any more made. And so it can transfer that information and communicate to the cell to make more or to not make that product just by releasing or not releasing that signaling molecule. Okay. And so, like I had mentioned, the sodium potassium pump is really important in terms of transporting sodium and potassium across the plasma membrane into and out of the cell as a result of helping the cell to maintain its specific volume. And as we know, cells are made up of a lot of water, and the amount of water that is inside of the cells is determined by the solute concentration. And so remember, if there's a lot of solute in a cell, water will go into the cell due to osmosis. Or if there's a lot of solute outside of the cell, water will go from inside of the cell out of the cell, causing the cell to shrivel. And so the sodium potassium pump really helps to maintain that cell volume. And so what happens is the pump gets rid of sodium ions from inside of the cell, and it builds up a concentration of potassium ions into the cell. And so the pump is moving 3 sodium ions out and bringing in 2 potassium ions for every churn of the pump. Okay. And so what this means is that if it wants to increase in cell volume, it needs more water in the cytoplasm. And so if it wants to increase in cell volume, it's not going to pump as much so that more sodium can build up on the inside, right? Because if you are kicking out 3 and only bringing in 2, you have a net loss of 1 to the outside every time, right? So your solute concentration is becoming lower inside the cell. So if you want water to come in, you need to build up that solute concentration so water will come in. Remember, water moves from high to low or from low to high solute concentrations. So if you want to decrease in cell volume, you want to lose water, then you pump a lot of those sodium ions out so that you have very little solute inside the cell, more solute outside, and so water will flow from inside to outside. And so again, this happens through the sodium potassium pump. So taking out 3, right, sodium ions bind, your ATP attaches, that phosphate causes that shape change, kicks out the 3 sodium, loses the protein loses those sodium binding sites. Now the potassium binding sites become available. The phosphate will kick off, and then it will cause the protein shape to change back, and those potassiums will come in. And so again, you have a net loss, right? So minus 1 ion every churn of the pump on the inside, and plus 1 ion for every churn on the pump outside. Okay. There are passive channels that allow sodium to come back in and potassium to go back out, because potassium is so we'll do the appropriate colors. Potassium is what? Colored green. So potassium's high on the inside, low on the outside, so it wants to move down its concentration gradient. And sodium is high on the outside and low on the inside, so it wants to move down its concentration gradient. So there are passive protein channels that allow that to happen, but this diffusion is fairly slow. Right? So it's fairly slow compared to the movement of these ions via the sodium potassium pump. Okay. And so when we're looking at the volume change of a cell in terms of how much water it takes in and how much water it loses, we refer to this as a tonicity. Okay. And so we basically can look at whether or not a cell is taking in too much water, like on this right hand example, where the cell would swell and it can burst. It's called a lysis, Lysis. It's called a cell lysis. Right? Or if there is a lot of solute on the outside, then the water will move out and the cell will shrivel. And then if the solute concentrations are equal, then you'll have an equal rate of water moving in and water moving out, which is on the left-hand side, so the cell will keep its volume, okay? So the sodium-potassium pump controls whether or not these three scenarios happen, okay? So if it pumps more sodium out, then water will move out. If it pumps more sodium in, then the water will move in. And then if it's, keeping the concentrations equal inside and out, then you get that left-hand example. Now, the concentrations of the solutes inside the cell versus outside of the cell is what we refer to as the tonicity. And the tonicity, which is this word here, is the relative concentration of two solutes in two fluids. So this is the key thing. It's two solutes and two fluids, right? So if you have a cell and that cell is surrounded by extracellular fluid, which is generally water, right? You have two fluids here in this example. You have the fluid on the inside, and then you have the fluid on the outside, okay? If the solute concentrations are equal, so if the fluid on the inside, let's say, is 10% salt, and the fluid on the outside is 10% salt, they're equal, so you get this scenario here where there is an equal rate of inward and outward diffusion of, where there's a equal rate of inward and outward movement of water in and out of the cell. This equal concentration of the solutes in both of the fluids is referred to as isotonic. So the extracellular and intracellular solute, that's the key thing, the solute concentrations are equal. And remember, the solute concentrations are the things that are dissolved in the water. Typically, it's like salt or sugar is what we refer to. And so the cells are going to maintain this normal volume in isotonic fluids. Now, we have these regulatory mechanisms that help keep that extracellular fluid isotonic, which again is that sodium potassium pump. Okay. Now, whenever you get variations in tonicity, right? So let's say we have our two cells. Draw two cells here, and we have our water, our extracellular fluid. Right, and so we still have our inside concentration, color. We still have our inside concentration versus our outside. Okay, so remember we said when those two concentrations are equal, they're isotonic. Now, whenever one concentration is higher, then it's hypertonic. I should change the wording on this because it's a little misleading. So this says extracellular solute concentration higher than intracellular solute concentration. This actually can be either way. Okay, so in this case, if the extracellular is higher, so if the outside, right, let's say is 30%, and the inside is 10%, then the extracellular is hypertonic, right? So it has more solute, more salt, so it's hypertonic. So the solution, the extracellular solution in this case, is hypertonic, right? Now, in this bottom scenario, you have your outer and inner. Let's say we switch the percentages. Okay, so the inter is higher than the outer, the extracellular. So now the extracellular is hypotonic. Okay. And so in these situations, the water is going to flow towards the more solute. So since the more solutes on the outside in this situation, water is going to flow out of the cell. Since there's more salt on the bottom situation, water is going to flow inside of the cell. Okay. Now, one thing I want to point out here is we talk about tonicity relative to the two fluids. Okay. So in this top example, right, the outside, the extracellular fluid is hypertonic. That means the intercellular fluid is hypotonic. Okay? Because the most is hyper and the less is hypo. And you refer to it as each solution. Right? So that means in the bottom example here, then, the outer is hypo and the inner is hyper. Okay. And so it's always relative to one another, and it's based off of that salt concentration. And so in this top example, because the cell is losing water, it could shrink and die. In this bottom example, because the cell is taking in a lot of water, it could swell or burst or undergo that cell lysis. So here we have a figure that's kind of showing those three examples, but here we have cells in beaker water. And you'll see these examples in your lab exercises too. So here, if we have an isotonic solution, so typically, excuse me, typically a cell has about, we're assuming 9 grams of salt inside the cell. 9 grams, 9 grams. And so if the water also has 9 grams, then they're equal, right? So it's isotonic. Now, if the cell has 9 grams and the beaker has 18 grams, then the beaker water is hyper. The cell water is hypo. Okay? So that means that it's going to lose water to the external environment. If it's pure water, it has very little salt, and so the cell has more salt. So now the cell is hyper, and the water is hypo, and the cell is going to take in salt or take in water, and so it could swell and burst. Okay. All right, so that does it for our second video, where we discussed transport of molecules, cell communication, and also how cells use that transport of molecules through, for example, the ATPase pump, the sodium potassium ATPase pump, to regulate cell volume, which is a function of diffusion and osmosis processes or osmotic processes. Right? And so what we're going to talk about in the final video is how cells get energy. So how they actually go about getting energy from molecules that we consume and put into our bodies. And so that last video, we're going to kind of focus on cellular respiration. So converting glucose into ATP using oxygen and also creating carbon dioxide. And so with that, we'll see you in the third and final video for chapter 3.